let's just jump right in. The living cell. I mean, it's one of the most complex, intricate little machines in the entire universe. So how in the world do we even begin to study something so microscopic, so full of moving parts? Well, that's exactly what we're going to break down. The incredible toolkit scientists have built over the years to see, to sort, and to finally understand the cell.
Okay, just think about this number for a second. 37 trillion. That's roughly how many cells make up you and me. So you can see the challenge here, right? It's not just about seeing a cell, it's about finding the one specific type you're looking for in that massive, massive crowd and figuring out what it's up to. And for that, you're going to need some seriously powerful tools.
And when I say powerful, I really mean it. Some of the tools we're about to look at can magnify things more than 1,000,000 times. We're talking about a level of detail that reveals a whole new universe, a world that was completely, totally invisible just a few generations ago. So, first things first. Before you can do anything else, you have to be able to see the thing you're studying. And for the first biologists, this was probably the biggest hurdle of all.
So what's the big deal? Well, if you were to put a living cell under a normal microscope, you'd see pretty much nothing. It's kind of like trying to find a perfectly clear marble in a glass of water. It's all transparent, there's no contrast, you just can't make out any of the important details inside.
So, scientists came up with two really clever ways around this. The first idea was chemical staining. You basically use dyes that stick to different parts of the cell, making them pop with color. The catch? The process to get the dye in there, called fixation, kills the cell. So you get a beautiful picture, but it's just a snapshot. The second way is much cooler in my opinion. It uses clever physics, these optical tricks, to boost the contrast in living cells. This lets you watch life as it's actually happening, in real time.
But that was just the start. The technology exploded from there. We got fluorescence microscopy, which lets you tag a single protein with what's basically a tiny glow stick so you can track it moving around. Then came confocal microscopy, which uses lasers to scan the cell and build these stunningly sharp 3D images, one super thin slice at a time. And to go even smaller, we had to ditch light altogether and start using beams of electrons. That's how we get TEM to see inside the cell and SEM to see its surface in breathtaking 3D detail.
Okay, so now we can see them. Great. But what if the piece of tissue you're looking at, say from your immune system, has dozens of different kinds of cells all mixed together? How do you pick out just the ones you want to study?
The answer is this absolutely incredible machine called a fluorescence-activated cell sorter, or FACS for short. The best way to think about it is like a super-fast, microscopic coin sorter. But instead of sorting pennies from dimes, it separates different cell types, like your T cells from your B cells, at a mind-boggling rate of thousands every single second.
So how does this thing even work? First, you tag your target cells with a fluorescent marker. Then, you get all the cells to flow in a perfect, single-file line past a laser. As each cell zips by, if it's one you've tagged, it'll light up. It fluoresces. A sensor sees that flash of light and instantly zaps the cell with a tiny electric charge. That charge is just enough to nudge the cell into a different path, right into a separate collection tube. It's just an amazingly precise way to get a pure sample of the exact cells you need for your experiment.
All right, next tool in the box. You've seen your cells, you've sorted them. Fantastic. But here's the thing. For most experiments, a few thousand cells won't cut it. You need millions, sometimes billions. You need to grow yourself an army.
This brings us to cell culture, and it forces a really big choice. On one hand, you can grow primary cells, which are taken directly from living tissue. These are genetically normal, they act a lot like they would inside the body, but, and this is a big but, they have an expiration date. They'll only divide about 50 times and then they just stop. The other option is to use a cell line. These are often started from tumors and they've been genetically changed so that they are immortal. They can divide forever, which makes them the workhorses of the lab. But you always have to keep in mind they aren't exactly normal cells anymore.
And when you talk about immortal cell lines, there's one that stands above all the rest: HeLa. Established way back in 1952, this was the very first time scientists managed to keep human cells dividing and thriving outside the body. It wasn't just a breakthrough, it completely revolutionized medicine.
But the story of HeLa is also a really powerful and complicated one. These cells were taken from a woman, a real person named Henrietta Lacks, without her knowledge or her consent. Her cells went on to save countless lives and enable breakthroughs we still rely on today. But her story has, rightly, forced us to have very serious conversations about ethics, consent, and recognition in scientific research, conversations that are still incredibly important today.
Okay, so we've got our army of cells growing happily in a dish. Now it's time for the final frontier, getting inside. How do we crack open the cell and start studying its individual parts, the nucleus, the mitochondria, the tiny ribosomes?
Now, this is a delicate operation. You have to break open that outer membrane. It's a process called homogenization. But you have to do it gently enough that you don't just smash all the tiny, complex machinery inside. Think of it like trying to crack a walnut perfectly so you get the kernel out whole.
So once you've cracked them all open, you're left with this soup of organelles. How do you sort them? You use a centrifuge, which basically just spins things really, really fast. By spinning that soup at different speeds, you can separate everything out by size and density. You start with a low-speed spin, and the heaviest stuff, like the cell nuclei, gets packed into a little pellet at the bottom. Then you take the leftover liquid, spin it faster, and out come the next heaviest things, like mitochondria. You just keep repeating this, faster and faster, until you've separated out even the tiniest little parts, like ribosomes.
And this is where it all pays off. After all that work, seeing them, sorting them, growing them, and taking them apart, we finally have the purified components of the cell. And now, we can start to ask some really, really specific questions.
Now that we have our purified parts, we can do things like a Western blot. So imagine you have a photo of a giant crowd at a stadium, and you have to find one specific person. That's basically what a Western blot does, but for proteins. It lets you hunt through a mixture of thousands of different proteins and find the exact one you're looking for. It's how we figure out what proteins are in which parts of the cell and what they're doing there.
And you know, all of these fundamental tools, seeing, sorting, analyzing, they all pave the way for modern marvels like CRISPR-Cas9. Because once we understood all the different parts of the cell, we could start thinking about building tools to edit them. And CRISPR is basically like a find and replace function, but for the genetic code. It allows scientists to go into a living cell, find one precise spot in its DNA, and change it. It's a total game changer for studying what genes do, and maybe, one day, for fixing the ones that cause disease.
And there you have it. It's a whole journey. From the basic challenge of just seeing a transparent cell, to sorting it out from its neighbors, to growing billions of them in a dish, and finally to unpacking their insides to decode their secrets. This whole progression, this entire toolkit, is really the foundation that almost all of modern biology and medicine is built on.
And that, of course, brings us to the future. We've gotten really, really good at taking the cell apart to figure out how it works. So the next great challenge is learning how to put it back together in new ways. What are the tools we're going to invent next, not just to study life, but to actually engineer it? That's the question that's going to drive science for the next generation.